Hi class, in this recording we're going to continue our discussion of protein synthesis, and we're going to talk about transcription in more detail. To review, transcription is when we take the message that's in the DNA, the genetic code within a gene, and we're going to copy that information down to make some messenger RNA. There's an enzyme responsible for this process referred to as RNA polymerase. Recall, ACE means enzyme. What does the enzyme do? It makes RNA polymers, and it'll bind to DNA and assemble pieces of messenger RNA. This process is going to require a start signal, and a lot of times the start signal is going to be a repetitive ta-ta-ta-ta-ta-ta sequence within the DNA that initiates transcription of a messenger RNA molecule. The RNA polymerase will have to open up the DNA helix and then read the bases of DNA from only one strand of the DNA, and it will make up the messenger RNA. When RNA polymerase finds a C in the DNA, it'll add a G to the messenger RNA, C's to G's. When it finds G in the DNA, it adds a C to the messenger RNA, G to C. But here's a key difference here. Messenger RNA, when it finds a T in DNA, adds an A to messenger RNA. RNA polymerase will add, when it finds a T in the DNA, will add an A to the messenger RNA. But here, when it finds an A in the DNA, instead of adding a T to the messenger RNA, it adds a U for uracil to the messenger RNA. So after transcribing our messenger RNA, the RNA polymerase will rewind the DNA helix, and the gene that's being transcribed can actually have several RNA polymerases transcribing at the exact same time. And we need a special sequence in the DNA that causes the RNA polymerase to detach and cease transcription. That special sequence of DNA is called the terminator. So now we have a piece of messenger RNA that has not been processed yet. It's in the nucleus, and we refer to this as pre-messenger RNA. This is the immediate product that is immature and has been produced by the RNA polymerase. And within the pre-messenger RNA, there are two kinds of segments that are called exons and introns. The exons are the segments of the pre-messenger RNA that are ultimately going to remain in place. The introns are the segments of pre-mRNA that are going to be removed from the messenger RNA. They're going to be edited out. These introns are not completely useless. They're typically going to be used to control protein synthesis, and also whether or not we keep an intron in is kind of variable. And this allows for us, by modifying which introns and exons are within the final messenger RNA, we can have slightly different variations of the same protein, all being coded for by the same gene. Those enzymes that are within the nucleus are going to remove the introns from the RNA and eventually splice only exons together. To help illustrate this process, I have this figure here. So we start with DNA. The DNA is going to be transcribed by RNA polymerase to make a pre-messenger RNA molecule. The pre-messenger RNA molecule is going to be edited, and it could be edited different ways. We could leave the A, B, C exons in place. We could leave the B, D, E, so on and so forth. So you can end up with slightly different variations of messenger RNA, all based on the same pre-mRNA molecule. And then those slightly different variations of messenger RNA are all going to produce slightly different variations of the same protein. This is referred to as alternative splicing, and gives us more bang for a buck. And this is how we can have millions of proteins within our body, but only thousands of genes. One gene can be used to make a variety of different proteins that are similar but slightly different from each other. Up next, we have translation. And this process of translation is when we go from a nucleotide to an amino acid, when we go from messenger RNA to the polypeptide of the protein. And we have three key participants or key characters during translation. We are going to have messenger RNA that carries the genetic code from the nucleus to the cytoplasm. we have transfer RNA that is going to be the interface, the molecule that is going to serve to as the bridge or the converter from nucleic acid to amino acid. It's going to carry amino acids into the ribosome, one amino acid at a time. And transfer RNA is a kind of nucleotide. It contains a region called an anticodon, which is 3 nucleotides that are complementary to the codon on the messenger RNA. And then we also have ribosomes. A ribosome is an organelle that is going to read the message contained within the messenger RNA. And ribosomes are made up of protein and nucleic acids. We find ribosomes free floating within the cytosol of the cell, floating or being attached to the rough endoplasmic reticulum or directly on the nuclear envelope. And as we look at ribosomes, to review from chapter 3, cell biology, they have a large and small subunit, and each subunit is made up of rRNA and several enzymes as well. Now, as we look at this messenger RNA molecule, it's always going to start with the leader sequence. This leader sequence is going to be the binding site for the small ribosomal subunit. After the small ribosomal subunit is bound to the messenger RNA, the large ribosomal subunit is going to attach to the small subunit. And then the ribosome is going to pull the messenger RNA through it like a ribbon, one nucleotide at a time, reading the bases. And it needs to just keep chugging along one base at a time until it reaches the start codon, AUG. And this is a key codon that codes for methionine, which is abbreviated as an M, ET for the 3-letter abbreviation of that amino acid. And it's really important that we work with start codons so we have the correct frame of reference. Here's one of my favorite sentences to illustrate this. We could have the fat cat. And what I like about the sentence is we have words made of 3 letters, kind of like codons are 3 nucleic acids long. And if we have the correct frame of reference, we can read it and say the fat cat, and we can have meaning, we have the correct meaning. But if we're off by a nucleotide, instead of reading the fat cat, I could read hef act, and it just doesn't make any sense anymore. And this is why we need that start codon, because once we reach the start codon, the ribosome will read every 3 nucleic acids downstream from the start codon, once it's achieved the frame of reference from the start codon. As we look at the tRNA molecules, that tRNA molecule has the anticodon down here that will match up with the codon in the messenger RNA. So in this anticodon of UUA would base pair with an AAU codon within the messenger RNA. And then this tRNA would have an amino acid attached right up here that would bring that it would be carried into the ribosome and added to the growing protein. After it adds that amino acid to the growing protein within the ribosome, the empty tRNA molecule is ejected from the ribosome, heads back into the cytoplasm, and then picks up free amino acid. This whole process of adding one amino acid to a growing protein is going to cost 1 ATP. In particular, the ATP is going to be used up when attaching the amino acid to the transfer RNA. As this process is occurring, as translation is occurring, we have 3 key steps: initiation, elongation, and termination. Initiation is when our small subunit is going to bind to the small ribosomal subunit binds to the messenger RNA, and then the large ribosomal subunit will bind next. And then we hit the initiator tRNA, or the start codon, and that brings in the initiator tRNA, which has a methionine amino acid. And then our large ribosomal subunit and small ribosomal subunit are able to move across our circular messenger RNA molecule. After initiation, we have elongation, and this is where most of the action occurs. We're going to have a tRNA molecule loaded with an amino acid enter into the ribosome, and then the anticodon of the tRNA base pairs with the codon of the messenger RNA, and that amino acid being carried will be dropped off and added to the growing polypeptide that's being squirted out of the top of the ribosome. And then our empty tRNA is released into the cytoplasm, where an ATP is used up to reattach another amino acid. And this process is going to continue again and again and again and again, making the protein the polypeptide longer and longer and longer until it becomes a protein. Eventually, we will hit a stop codon, and it's that stop codon that is going to cause this process to terminate. Speaking of termination, the termination phase of translation is when we reach the stop codon. And that stop codon is going to cause the large and small subunits of the ribosome to separate and protein synthesis to stop. This process of translation can be quite rapid. We can have one messenger RNA with multiple ribosomes, typically 10 to 20 ribosomes. So shown here in the bottom, in red we have the messenger RNA molecule, colored blue we have ribosomes, and then colored green, all these little green squiggly lines are polypeptides that are being formed by individual ribosomes. So this process can allow for lots of simultaneous translations to occur at the same time. We can have up to 300,000 identical messenger RNA molecules within the cell at the same time, and each of those messenger RNA molecules that are in the cell, those 300,000 can have 10 to 20 ribosomes all going through translation. This means that ultimately we can make over 100,000 protein molecules per cell per second. We have an amazing ability to pump out protein. Here is another fantastic review video. If you get a chance, please watch it. I can't play it in this recording due to copyright issues. YouTube tends to frown on YouTube videos being hosted within other YouTube videos. If you have any questions or comments about this content, excuse me, I got ahead of myself here. So, to review on how we make proteins, we start with the DNA double helix. The triplets in the DNA double helix are going to be transcribed to make a messenger RNA molecule. And then within the messenger RNA molecule, once we have the start codon read by the ribosome, we will then base pair the codons with the anticodons in the tRNAs. The tRNA molecules are then going to bring in different amino acids, and those different amino acids are going to be added to the protein or polypeptide as the process of making a protein proceeds. Once the ribosome hits the stop codon, the large and small subunits dissociate, and we stop adding amino acids to the growing protein. Notably, when we hit start, the start codon corresponds to methionine. The stop codon does not have an amino acid that corresponds with it. So, if you have any questions or comments, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.